and welcome to your next video lecture on mitosis. So we've talked a little bit about mitosis in class. We've talked about how we're changing our cells and we're duplicating them. And we go from a parent cell to a daughter cell. So now we're going to explore a little bit more about mitosis. So why do our cells divide? Our cells divide because we need to grow. Remember, that's a characteristic of life. You are constantly growing. Even if you're not getting bigger or smaller, you're growing because you're replacing the cells that you already have. Because remember, 300 million of our cells die every single minute. We're going to develop. Think about a baby needs to develop into adulthood. And so we're going to use this process in order to develop into that adult. And if you've ever skinned your knee, you know that eventually that skinned knee has new cells on top, that new skin that forms. And so what we use that for is repair. So we have two essential features of cell division. We transmit a complete copy of the genetic information, or the DNA, into a new cell. And we transmit materials necessary for the cell to survive and use genetic information. You might recall that we have that information in the DNA, and we're going to learn in a couple weeks about transcription and translation and what that is necessary for. So we're talking all about this DNA. And remember, the DNA is in the nucleus, and it has to be able to be replicated in order to go through mitosis. So what happens to all that DNA? Well, let's explore that. Remember about interphase. During interphase, DNA molecules are extended in an uncondensed form, which is called chromatin. Remember I told you to think about a ball of yarn or a plate of spaghetti. It's really loose, it's unwound. It can be very, very messy as well. So once we get into the cell cycle, though, we're going to condense that chromatin into chromosomes. So the DNA molecules condense to form our chromosomes prior to division. And do you remember what phase that happens in? If you said prophase, you would be correct. So pat yourself on the back. Chromosomes are DNA that is tightly wound around proteins. They're connected by a centromere. So that little dot we see in the center is going to be our centromere. Now, go ahead and copy this figure down. That first one you see, that number 1, is going to be the DNA molecule, which you know this, go ahead and write that down. If you don't, it's a double helix. DNA molecules are wrapped around the proteins that you can see in number 2. So if you look at these turquoise balls here, well, those are proteins, and the blue is our DNA, and we're just wrapping that around our proteins there. And number 3, we actually have unwound DNA, but you can start to see that centromere forming when it's winding up. So this red dot here is going to be our centromere, and it's all unwound. Now, we can see it starting to condense into the chromosome in number 4. And it's not totally wound, it's not totally condensed into a chromosome, but that chromatin is becoming a chromosome, and we can see it getting tighter. And lastly, number 5, we have our chromosome. So let's go ahead and move on. I want you to copy this figure as well into your notes. You can see that we start out with a DNA double helix here. We wrap around those proteins like we just saw. And those proteins are actually called a histone. I would make a good note of that in your notes. And then we're going to go into nucleosomes. You do not need to know what that is. But let's look all the way at the bottom here. At the bottom here, it has a chromatin loop. You don't necessarily need to know that, but what you might want to know is this unwound portion of DNA is our chromatin. So chromatin is fairly important, and when we wind it all up, we get a chromosome. So remember, that's connected by a centromere. Okay, so what is mitotic cell division? We've talked about mitosis, we've talked about all kinds of things. Mitotic cell division is the division of a somatic cell. So a somatic cell is going to be your body cells. And this only happens in eukaryotic organisms. Somatic cells do not occur in prokaryotic organisms. A single cell divides into two identical daughter cells. So nothing will be different about these new cells. So as you can see here, this is the end of mitosis, but we're going from one cell to two cells, which is very important to note. Go ahead and, if you have room, put this in your notes as well. So mitotic cell division, we actually have two major processes that we go through. And this is the phase of mitosis of the cell cycle. It is not the entire cell cycle. So mitosis divides the nucleus and chromosomes. Cytokinesis divides the cytoplasm. And what's actually happening in cytokinesis is we're getting those two separate cells. Now we can go through mitosis over and over and over again, and we cannot go through cytokinesis, and then we have all kinds of, we're duplicating our nucleus. So we might have a cell with like 50 nuclei, which I find really cool. Okay, so just remind yourself that mitosis has 4 phases, and they are prophase, metaphase, anaphase, and telophase. Remember in class we talked about PMAT. Now telophase includes that process called cytokinesis, which is the division of the cytoplasm. Very important if you're going to have a new cell, but we don't necessarily have to go through it. So prophase. Now you're going to want to copy all of these things down, so listen very, very carefully. Chromatin condenses into chromosomes, and so we now have those tightly wound chromosomes. And we get spindle fibers that are going to form and attach to our chromosomes. Now, those spindle fibers come from an organelle in the cell. Do you know what it's called? If you said centriole, you would be correct. Pat yourself on the back again. The nuclear envelope now breaks down. So we had this nucleus. It had a very structural envelope around it, and now it's going to begin to break down because we need to use that whole cell in order to divide. So let's start looking at what this is. So in this photo, you can see that this cell is going through prophase at this time. We see portions of the unwound DNA, but it's all condensing into chromosomes. And as you can see here, we have centrioles, and these orange squares represent our centrioles. And you can see these fibers coming off of them. Well, they are spindle fibers, and they're eventually going to attach to our chromosomes. And look at this, our nuclear envelope. You see all those dots around it? Well, it's starting to dissipate because we need that whole area in order to go through mitosis. Okay, so the next phase, metaphase. Remember, I told you M stands for middle. So remember, metaphase, middle. Very, very important to remember. So here we have our chromosomes all condensed now. We've got that chromatin into the chromosome, and we're going to align along the equator of the cell. As you can see, this is our cell here, and they all line up perfectly in the middle. So that's a very, very short phase of mitosis. And once we have lined up there, we go into anaphase. Anaphase is where we're pulling our chromosomes apart into sister chromatids. And remember when we talked about interphase, that S phase is where we actually duplicate our DNA. Well, until you hit after anaphase and until you hit the S phase, you do not have chromosomes, but you have chromatids. Once you duplicate that DNA, you then have chromosomes. So, spindle fibers then attach to the centromeres, and they shorten and pull the chromatids towards opposite poles. So we basically just break it apart. And remember when we looked at the cells, it kind of looked like this. So we had gone from metaphase, which was this. We're now pulling them apart, and these are our sister chromatids. So they're being pulled to opposite ends. And then finally we free those spindle fibers, we lengthen and push poles of the cell apart. So we lengthen and we push them to the poles. Okay, so this is a picture of anaphase. I know I just showed it to you with my fingers, but this is a better picture to have, and obviously copy this in your notes. I know you have notes from class earlier with anaphase, So if you feel like that's sufficient enough, that is fine. Okay, so telophase, our last phase. Telophase, the spindle fibers disintegrate, the nuclear envelopes form around both groups of chromosomes, and the chromosomes revert into their extended state, so they become chromatin again. So remember, we had the sister chromatids. Technically, we now only have chromatids. We do not have chromosomes. And they're going to go back to, so they're going to unwind, uncondense, and they're going to go into that chromatin form. And then lastly, we have cytokinesis occurring. And that's enclosing each daughter nucleus into a separate cell. Now remember, I said we could go through telophase over and over, or we could go through mitosis over and over and over again, and we would just have multiple nuclei in one cell if we did not go through cytokinesis. So it's very important to recognize you can go through mitosis without going through cytokinesis. And lastly, this right here is going to be a cleavage furrow. A cleavage furrow only occurs in animal cells, and that cleavage furrow is when you are pinching in to create the new cell. So we've gone from one cell, which is our parent cell, to 2 daughter cells. Okay, this is your video on mitosis. You also have a video on meiosis. We will see you in class.